Assalamu alaikum students. Today we will start Unit 3, The World of the Living, Chapter 8, Heredity in Organisms. Introduction. All living organisms grow, multiply and produce one of their kinds. Life exists on earth only because all living organisms can reproduce. If a given species is unable to do so, it becomes extinct. We know that all living organisms are made up of cells. What occurs at the cellular level during reproduction? How does a unicellular or a multicellular organism produce offspring? In this chapter, we shall find answers to the cellular fundamental mechanism of reproduction. Cell division. The process by which cells produce their exact replica or duplicate is known as cell division. Through cell division, new cells originate from pre-existing cells, and in this way an organism grows. All cells grow through a cell cycle during their life before they divide to form new cells. The cell cycle is an ordered set of events resulting in the growth and cell division of cells into two daughter cells. Cells divide to grow, increase in size of multicellular organisms, repair and replace the damaged cell parts. Life cycle of cells involves two main events: interphase and cell division. There are two types of cell division: mitosis and meiosis. The cell division associated with the sexual reproduction is called meiosis, and the cell division associated with the growth and cell replacement repair is called mitosis. Mitosis. During mitosis, a cell duplicates one of its contents, including its chromosomes, and splits to form two identical daughter cells. These daughter cells have the genetic material of the parent cell, that is, a complete set of chromosomes, organelles, and cytoplasm. Various stages of mitosis are prophase, metaphase, anaphase, telophase. Prophase. It is the first stage of mitosis. During this phase, the nucleus fades and chromosomes become more coiled. Each replicated chromosome comprises two sister chromatids, which are joined to each other by the centromere. You can see a diagram in front of prophase. Metaphase. During metaphase, spindle fibers align the chromosomes along the middle of the nucleus in one plane, that is, equator of the cell. This line is referred to as metaphase plate. This alignment helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. The nuclear membrane completely disintegrates, and centrioles are arranged in pairs at the opposite poles. Anaphase. Anaphase is marked by the contraction of spindle fibers, leading to the splitting of the centromere. The sister chromatids, daughter chromosomes, move apart towards the opposite ends of the cell. Telophase. The daughter chromosomes arrive at the poles, and the spindle fibers that have pulled them apart disappear during this phase. Nuclear membrane begins to form, nucleus reappears, and the chromosomes become less tightly coiled. This is followed by cytokinesis, division of cytoplasm to form two daughter cells. Significance of mitosis. Mitosis becomes significant for the following reasons. Mitosis forms two daughter cells which have same chromosome number and same genetic material as the parent cell. daughter cells formed from mitosis are genetically identical to their parent cell and hence no variation is introduced. This results in genetic stability within the populations of cells derived from parental cells, as in a clone. The number of cells within our organisms increases by mitosis. It forms the basis for growth. If mitotic division goes uncontrolled in any part of the body, it results in the formation of malignant cells. These cells continue to divide, resulting in the formation of malignant tumors. This condition is called cancer. Meiosis. Meiosis is a type of cell division that occurs only in the reproductive cells and hence that is known as germ cells division. It is a two-step process divided into first meiotic division or meiosis I and second division or meiosis II. In both the meiotic divisions, nuclear division can be distinguished into 4 stages: prophase, metaphase, anaphase and telophase. Meiosis reduces the chromosome number by half. In the case of humans, number of chromosome decreases from 46 to 23. Meiosis I. The first meiotic division or meiosis I is commonly described as reductional division. The chromosomes of the parent cell segregate, producing 4 daughter cells. Phases of the meiosis I are discussed. Prophase I. The chromosomes get shortened and become visible as single structures. Homologous chromosomes pairs and form synapses. Synapses is the process of linking of the replicated chromosomes together. Non-sister chromates of the paternal and maternal chromosomes overlap, resulting in crossing over. The chromosomes condense again. Metaphase I. Bivalents. The bivalents, each composed of 2 chromosomes, 4 chromatids, move towards the equator of the cell and come to lie in 2 parallel metaphase plates. Anaphase I. Homologous chromosomes, each with 2 chromatids, move to opposite poles. Each of the daughter cells now has at least 1 chromosome of the homologous pair. Telophase I. The chromosomes at each pole uncoil and elongate to form the chromatin. A nuclear membrane is formed at each pole, resulting in the formation of 2 daughter nuclei. Meiosis II. Meiosis II is similar to mitosis. This division is primarily meant for separating the 2 chromatids of each chromosome. The chromatids of each chromosome are no longer identical because of crossing over. Meiosis II also has a karyokinesis, splitting of nucleus, and a cytokinesis, splitting of cytoplasm. Meiosis II can be distinguished into 4 stages, namely prophase II, metaphase II, anaphase II, and telophase II. Significance. Meiosis becomes significant for the following reasons. It provides a chance for the appearance of new gene combinations as a result of crossing over. This allows for random choices between chromosomes. This is why children look different even though they have the same parents. Also, just the fact that you are mixing your DNA with your partners increases genetic diversity, allowing better chance for survival to new environments. This division is necessary for the formation of gametes in animals and spores in plants. Heredity and variations. Heredity is the transmission of physical or mental characteristics from parents to offspring, that is, from one generation to the successive generation. Although offspring resemble their parents, they are not identical to them. The differences shown by the individuals of a species is termed as variations. It represents the characteristics that distinguish between two individuals of the same race or between the offsprings of the same parents. These characteristics can be called traits. Gregor Mendel was the first to study the transmission of traits from parents to offsprings. Chromosomes and genes. In the nucleus of each cell, there are a number of thread-like structures called chromosomes. Each chromosome is made up of very long molecule of DNA. The DNA molecule is responsible for the information storage, transmission, and expression of hereditary traits in organisms. There are certain factors which control the inheritance of traits. These factors are known as genes. Genes are therefore the units of heredity and are made up of specific DNA sequences. It is the genes on your chromosomes which determine all sorts of things about you, like color of your eyes or hair, shape of your nose, etc. Humans normally have 46 chromosomes in each, forming 23 pairs. Inheritance. Genetic inheritance is the transmission of genetic material from parents to offspring. Children inherit traits, disorders, and characteristics from their parents. Some traits involve single gene, while others are complex, involving many different genes. Traits such as blood type, eye color, hair color, and taste are controlled by a single pair of genes each. Some traits are found to be determined by multiple genes as well as environmental factors. For example, height seems to be controlled by multiple genes. Some are tall genes and some are short genes. A child may inherit all the tall genes from both parents and ends up taller than both parents. Or the child may inherit all the short genes and be the shortest in the family. More often, the child inherits both tall and short genes and ends up about the same height as the rest of the family. Good diet and exercise can help a person with short genes end up attaining an average height. Sex-linked inheritance determines the gender of a person. Genetic disorder. You must have seen doctors asking patients, does their parents, father, mother, or grandfather have the same symptoms? Just in the case of diabetic patient, they are often asked about their inherited disease. A particular disorder might be described as running in the family. If more than one person in the family suffers the same condition, some disorders that affect multiple family members can be inherited, that is, that can be passed down from parent to children. When the chromosomes are incorrectly distributed in the egg or sperm, one cell may get 2 copies of a particular chromosomes, which is not normal, while another gets none. This leads to genetic defects when gametes fertilize. For example, if a sperm with the extra chromosome fertilizes an egg with the normal chromosome number, the resulting zygote will have 3 copies of one chromosome. Variation in the number or the size of chromosome in a species results in genetic disorder. Thank you.